So now it's my pleasure to introduce Dr. Jeffrey Hubbard. He earned his PhD in fundamental neuroscience in sleep neurophysiology at the University of Strasbourg. And he's now a translational researcher specialized in the regulatory mechanisms of sleep and waking behavior and clinical sleep disorder. And today he'll talk about the opposing forces of light and darkness on sleep and waking behavior, lessons from nocturnal and diurnal animals.
So thank you so much again to the committee for giving me this opportunity to talk. So as Virginie said, I have spent my research career kind of looking at different species across from small little rodents, which I'll talk about today, all the way up to clinical sleep disorders. So what I wanted to do today is to take a few steps back down the staircase of evolution to these smaller animals to give you an idea of what we use these animals for in laboratory research to better understand kind of the fundamental processes of how light is interpreted by the brain and how this affects sleep and waking behavior. So today I'm going to talk specifically, I want to talk about the direct effect of light, and I'll tell you what I mean by this. Renske already mentioned this a bit. And then how can we isolate this direct effect in laboratory mice? So in a controlled setting, what would we then observe in diurnal rodents as a consequence? So just very briefly, how sleep and waking are kind of thought to be regulated, at least at this point, is we think about these two processes of their circadian and homeostasis. So the circadian process is this idea of gating sleep and waking based on a temporal niche for the animal, so that some animals are more day active, some animals are more day active, some are more night active, some are somewhere in between. Of course, behavior can override this, as we know from sleeping during the day or staying up the entire night. As both of the previous speakers said, this is controlled by the central clock in the hypothalamus, the suprachiasmatic nucleus. We have also this other process called the homeostatic process. This is very broadly speaking, it keeps track of sleep propensity. It's an idea represented by an hourglass there. It can be reflected in low frequency brain activity during sleep and other physiological signals. But I would also like today to talk about something called the direct, let's say the direct effect of light, or a non-circadian process. You can think of this as a bit like a flashlight at night. So obviously the pupillary reflex is a very common thing that everyone has noticed. If you go into a bright area, your pupil will constrict. Vice versa, if you're in a dark area, it will slowly expand to allow more light in. This is important for melatonin suppression as well, which Virginie mentioned. We do also know that, and I will discuss this today, it can be also affect acute induction of sleep and waking phases. So of course, again, if you shine a flashlight in somebody's eyes while they're sleeping, they will wake up to certain to a certain extent. And this idea is supported by studies not only in model organisms like rodents, but also in human research and clinical studies as well. So how is light interpreted by the brain? I know we've already discussed this twice today, but just to give you an idea, again, what I'm going to discuss, so this non-image and image-forming cells that Renske mentioned and Virginie are located, and let me just turn on my laser pointer, located in these melanopsin or OPN4 intrinsically photosensitive retinal ganglion cells. We also know that rods and cones are important for this, although to a much less strong extent, they're more indicated for image forming. The melanopsin cells themselves in the retina, they project to a lot of parts of the brain. Here's just a very few examples. One, the superior colliculus, so very important for light information to pass to areas of the brain involved in orientation, but also to the hypothalamus to control our eating behavior and then also sleep and waking. So why do we use animal models? This is always a discussion, of course, in research. one of the reasons we do it is to have a completely controllable environment. So in terms of light, it would be about manipulating the amount of exposure the animal has, but also the intensity. If we want to do experiments where we shut light on and off, it's much easier to do this in a controlled environment. Of course, we can also measure things with the feeding behavior if we want to control the amount of food or the type of food that they get, how this influences their behaviors, we can do this. Second thing is that it allows us to do long-term physiological capture. So in the case of what I'll discuss today are multi-day recordings of sleep and wake behavior. The most obvious reason that we use this in a broader sense in biology and neuroscience is that we have the opportunity to add and remove genetic, we can use genetic tools to add or remove genes to the animals in order to see how they react. And today, for example, I'll discuss what happens when you remove the gene that encodes for melanopsin. It also gives us evolutionary insight. So again, looking at these different temporal niches, why these animals evolved in a certain way and how this interacts us in their brain. I will give you a spoiler. For example, we still don't really know why some animals are nocturnal and diurnal because the light going into their eye is the same light. So we're still working on it. Hopefully in some years we'll figure this out, but we have some hypotheses, but we don't know yet. So also, finally, what's very important is the translational impact. There's very often discoveries that were made in animal models led to changes in sleep medicine and chronobiology, and even certain ideas about public health recommendations come from these original models. So we have talked multiple times now about melatonin. And it's very important, certainly when we talk about chronobiology, light exposure, circadian research. The problem with mice in the laboratory is the majority of them do not produce melatonin. This is because some mutations that exist in the most commonly used models that will modify melatonin synthesis, but also it's quite possible that animals which were naturally more geared towards breeding at certain times of the year under certain light dark conditions had mutations in melatonin production. So we kind of force the mutation into the animals. And then we do also have some strains which are used less optional for example, the CBA strain and the C3H strain of mouse, they do produce melatonin. Just to show you what this looks like. So the normal C57 Black 6 mouse, which is what is used in most cases, they have no melatonin production over the night. However, we talk about mice being nocturnal, but their melatonin peak is still during the night when they are active. So we also don't know what role melatonin plays in nocturnal animals versus what we presume the role it plays in human beings. So how do we measure sleep and waking in rodents? We use something very similar to what is done in humans that Rensky mentioned, so with electrodes. However, because the mice won't sit still, we have to put the electrodes directly onto the animal. We record with a system that allows us to capture brain activity and muscle activity. So here we can see a waking signal, for example, very kind of high frequency moving around, a lot of movement here on the EMG. This is the muscle activity. When the animal goes into normal sleeping, we have much slower brain activity, not a lot of muscle movement. And then rapid eye movement sleep. So we don't know whether the animals are dreaming during the sleep, but in any case, they have an active brain, but a relatively suppressed muscle tone. So experimental evidence in mice, for example, on the role of light and dark and sleep and waking, I just want to briefly make some points that mice in general, there are some mice that are more geared towards diurnal behavior, but in general, mice are preferentially active during the dark. It doesn't mean that they won't sleep in the dark. It just means the majority of their activity takes place in the absence of light. We also know that acute pulses of light, so turning a light on, for example, in the middle of the night, will induce sleep in these animals. And we know that this is done through melanopsin phototransduction, so through these cells. But we also know that light exposure can exert a sustained effect. So it's not just these single small pulses of light, but also the exposure over time, which will affect the behavior of the animal. So this was back in 2009, this data was initially published that showed what would happen if you remove the melanopsin protein from the animals, so rendering these cells in the retina inert. And what we see here are images from the suprachiasmatic nucleus of mice with and without melanopsin retinal ganglion cells. And we see in mice, these OPN4 minus minus, that there is a reduction in the amount of cells which are activated by a light pulse. And when we look then at the sleep in these animals, we see that in fact these mice here in the light period where they are more likely to sleep versus the dark period, in the black here, the melanopsin knockout mice, they are sleeping around 1 hour less during the light period, which is not recovered during the dark period. So there is a sustained reduction in sleep driven by the lack of this protein in the retina. So what pathways might be responsible? Both the previous speakers discussed the hypothalamus. Our current framework for a night active or nocturnal species is to look at this, for example, the ventrolateral preoptic area, which we know is very important for sleep induction, that this flip-flop model kind of exists, where as you have increase on the pressure to sleep, you have a decrease on the pressure to stay awake, and vice versa. We know through the OPN4 cells that these go through the SCN. We can see this on the anatomy. So we know that there is a direct innervation from the retinal hypothalamic tract into these brain areas, also to the end. But we don't really know how this feedback loop works. So how might it work? Does the SCN relay these effects as a secondary function? So it seems pretty clear, yes, but to look for experimental evidence, we kind of have to find a way to dissect this. Is the melanopsin-based photic input significant enough or sufficient to drive this sleep-wake cycle? So if you remove the circadian rhythm, will the animal still sleep? We know, for example, Virginie showed the data from the cave experiment. This is where you remove the light influence, and then you see the circadian influence still shows the persistence of a clock that kind of organizes sleep-waking behavior, even though it is free running. But then finally, can we separate then this circadian and direct influence of light? So what we did is we took mice. On the top here, we have mice similar to the ones I just showed you, so lacking melanopsin, and then normal mice. And in the bottom, we have mice where we removed the suprachiasmatic nucleus. So we removed the circadian function, as well as this pathway for the melanopsin information. And then we moved the melanopsin in addition to removing the SCN. And what we found is when we subtracted the amount of non-REM sleep or sleep between the light and dark periods, we saw this progressive decrease depending on what was removed. And the most interesting finding, for example, so again, confirmed that they slept 1 hour less, but that the mice without a circadian rhythm still sleep longer during the light period. So this is a proof of the fact that there is this direct effect of light, which is promoting sleep, regardless of a functioning clock. But once you remove both of these things, the animals become arrhythmic, meaning that they sleep the same amount during the light and dark period, and they are awake the same amount, as though there's no light going into the brain to change behavior. So what can we do with information like this? So what we did is we took this information and we tried to find a model representation of what all of these differences might mean. This is not so important, but what it does is it allows us to come up with this framework where we identify that the circadian rhythm by itself under this normal light-dark exposure is responsible for about 55% of the sleep-wake period behavior, whereas these sustained direct effects of light, so the exposure over 12 hours of light and darkness, that this is around 45%. Now, these are general numbers. This does not mean that it would work in every scenario or that it would even be translatable to other species. But the next thing we wanted to do is see, can we use these numbers, very simple 55 and 45%, and shorten the photo period? So to run an experiment that would mimic winter light exposure in northern and southern extreme latitudes. So we shorten the period to 8 hours a day. And what we find is that just using these two numbers, we're able to more or less model it doesn't account for all changes, but more or less model the amount of sleep. And there we see that it's fairly good at adjusting for this reduced amount of light exposure. So, but one thing we wanted to do is maybe come up with something a bit more complicated and a bit more maybe important. Now, everybody probably has experienced jet lag to different levels. Of course, I know that Virginie and Rensky both lived, or Rensky still lives in California. They're both from Europe. I came from California and now live in Europe. I've certainly been on the plane many times with these 9-hour jet lags or longer. The basic idea with the jet lag is that you end up with two clocks that are out of sync. And we know that this causes disturbed sleep. We know that it causes daytime fatigue, difficulty concentrating, and mood changes as well. So, can you jet lag a mouse? Now, short of putting them on an airplane with suitcases and making them wait in security, maybe we don't have the same stress level with the mice as with human beings. But what we can do is we can shift their photo period. So we can substitute or kind of simulate an 8-hour westward jet lag just by shifting the start of the light period. And what this gives us here on the bottom, you can see some actimetry from one animal, similar to what Rensky showed in humans with the ActoWatch. We see that when the jet lag occurs, we start seeing this kind of shifting that occurs. And then after a few days, it seems to realign to this new photo period. So how do we apply the model to something like this? So we did some calculations, some fitting, and we found that a 2.2-hour shift per day could be applied to the circadian effect, meaning that there was this sine wave that was moving and it was kind of shifting a little bit every day. And what this gave us here under our baseline, we see our prediction versus our circadian and sustained direct effect model. What we saw is that if we slowly move the circadian curve, for lack of a better word, after about 4 or 5 days, it starts synchronizing. So this is how the model looks. Now, again, it's hard to fit this directly to animal behavior. There were 8 mice in this experiment, but it more or less kind of fits over the idea. So, and when you look at broader kind of light dark differences rather than these 3-hour blocks of time, we see that the model is fairly close given the variability in the data. So maybe the feeling and the resetting after jet lag, and this is not only a combination of a circadian effect, but is also a light exposure effect. And we certainly know that this is told to us to get light exposure at the right time. And this also helps with our eating behavior and things like this. So just to summarize briefly, in these nocturnal animals and the mice, so light influences sleep not only through the circadian timing, but also through this direct effect. I call it a non-circadian response. I'm not trying to be provocative, just to say that it's something that seems to persist outside of the circadian rhythm. And that even in the absence of the SCN, light will modulate sleep. Although an intact circadian system is necessary to have this rhythmic behavior or the sustained sleep-wake behavior. But understanding these effects may help us to predict how people react under these irregular lighting conditions, especially if they're shifting back and forth very quickly, either through travel or through shift work, So now the last few slides I wanted to focus on are in the diurnal rodents. So I had the opportunity during my PhD to work in the Department of Integrated and Cellular Neurosciences. It's actually where I met Virginie when she was a master's student there. And we had this animal called the Arvicanthis ansorgei. So the Arvicanthis ansorgei has a more general name, the Sudanian grass rat. However, these are from Mali. So they were captured about 20 years ago by researchers in the department, and they have at this point been bred for more than 20 years in captivity. But let's say 10 years ago, when I was using them, 12 years ago, they were a bit more wild. But they're fairly small. They're not quite the size of a full rat. So they're certainly much bigger than a mouse, but they're very nice. You can see they have this nice fur. And they've been used in a lot of chronobiology studies, especially to look at seasonal reproduction, also to look at retinal function, and how they, as a diurnal animal, how they discriminate with non-visual function in different light intensities. But what I was interested in as a sleep researcher was what did it look like, their sleep and waking behavior. So along with people in the lab, we recorded them for the sleep and wake behavior. And the first thing you might notice in blue here, we see the wake, we see a lot of activity that occurs between these light and dark transitions. So that has this crepuscular behavior. Now, this makes sense, the sunrise and sunset periods in the wild, that this animal, which is much less adapted to a laboratory compared to a mouse, which has been bred for 90 years in a laboratory. But we also see that there is more sleep that occurs during the dark period. So the first thing we wanted to do, focusing on light, is what happens if we remove the light. So you remove the light, it has still this strong crepuscular rhythm, and it has still this, what looks like a non-REM increase, sleep increase during the subjective dark period, the second half of the night. However, if you actually look at these differences and these 12-hour differences, we see that there's quite a strong difference, reduction in the non-REM sleep when you remove the light. And the same is the case with waking, although to a lesser extent. So then what happens if you try to induce waking by light? So to turn on the light in the middle of the night, what happens to the animal? Well, what we saw is that if you turn the light on 3 hours after the light has been turned off in the laboratory, we see this induction of waking that starts around 5 to 10 minutes into the hour and is sustained over the hour, but then takes a bit of time to go back down. This was also paralleled in high frequency brain activity that we normally associate with exploratory behavior, cognitive processing, as much as these animals have. And what was interesting then, if we look at this same brain activity under these sustained conditions, what we see is, well, there is always this strong activity at these light-dark transitions and dark-to-light transitions, which is reflected but during the subjective light period of the constant darkness, so when light normally would be, we do not see this high-frequency brain activity, whereas we do see it when the animal is exposed to light. So this seems to then imply again that there is some light exposure-dependent behavior. So what if we did this back and forth? Every hour we change either light or dark, light or dark. So interestingly, what happened is we see that there's an induction in waking activity. So when the brain is more active, although it is a bit delayed. However, it is fairly opposite to what we would see in a mouse, whereas in a mouse, we see this induction of waking by a dark pulse. So I would say it's a good diurnal model. It is not a model for human beings directly, but neither are mice models for human beings. They have less consolidated sleep than human beings do, but it does show that there is an increased wake behavior in response to light. You see that there is this light-dependent activity, increase in activity in the brain. And it sustainably increases wakefulness even during times of the day when they normally would be asleep. And then finally, that the temporal niche probably dictates the function of the environmental light. So just to summarize quickly what I was speaking about today, we have a lot of artificial light exposure. As Rensky mentioned, as Virginie mentioned, there is a lot of light that we're getting at weird times. We know that this probably contributes to insomnia and early arousals in certain cases. Of course, with jet lag and shift work fatigue, where light cycles are being changed constantly, and also to mood disorders and what we could say is cognitive fog, some kind of fogginess. But in conclusion, I would like to say light regulates sleep and waking not only through a circadian clock, but also through this acute and sustained effect, which seems to operate in parallel to the circadian rhythm. The neurobiological rhythm mechanisms are fairly clear. This seems to be very strongly regulated by melanopsin. There are species-specific responses, which we always need to keep in mind. And there is variability. These are laboratory animals, so there's a lot of variability in the wild as well. But finally, research from these animals can kind of help us identify what this direct impact of light has on behavior and lead to more general ideas, hopefully, about how we interact with light as human beings and how this may affect healthcare and other outcomes. So I want to thank again the organizers and of course my colleagues at the University of Strasbourg and the University of Lausanne